Good afternoon, everybody. Every trait, whether physiological or pathological, has a genetic basis, and this can be attributed to the four-letter alphabet DNA. You may wonder that there are only three letters in the word DNA, but this complex molecule is made up of sequence of bases called adenine, represented by A, thymine, represented by T, guanine, represented by G, and cytosine, represented by C. The characteristics of any individual are determined by the sequence of these bases across the DNA molecule, and any change in the sequence of these bases can not only change the characteristics of the individual but can also alter the susceptibility to disease. Today, I am presenting the findings of my systematic review titled Polymorphisms of Immune Response Genes and Early Childhood Caries. Systematic review, gene-based, gene cluster, and meta-analysis. These are the contents of my presentation. As we all know, ECC is a complex disease, implying that both the genetic and the environmental factors play a role in its etiology. The role of environmental factors, such as the flora and the diet, has been well established. The advent of molecular biology has paved the way for research into the genetic factors that influence the susceptibility to one of these most common conditions, which is prevalent in almost about 50% of our population. As stated earlier, single nucleotide polymorphisms or SNPs refer to a change in the nucleotide sequence across the DNA, and these SNPs are the most common variations in the human genome. SNPs of the genes regulating the immune response can alter the colonization of the mucosa by the bacteria and affect susceptibility to disease. The review of literature has revealed that complement activation, humoral immune response, regulation of cytokines and chemokines, and cellular immune response are all regulated by immune response genes, and variations in these genes can affect these processes. As this slide depicts, the various polymorphisms can alter the susceptibility to early childhood caries. Genotypes of certain polymorphisms being protective against ECC, and genotypes of other polymorphisms increasing the susceptibility to early childhood caries in the analyzed populations. Hence, the aim of this systematic review was to analyze the association between genetic variants of immune response genes and early childhood caries. This systematic review was registered with Prospero and was conducted as per the PICO framework. P for population, that is children up to 6 years of age. E for exposure, that is variants of immune response genes. C for comparison, comparing it to children without the polymorphisms of these genes. And O being the outcome, that is early childhood caries. An extensive search was conducted across the electronic databases and also in genome-wide association databases. This search was complemented by hand search to identify pertinent literature on this research question. This search across the databases led to selection of studies, and data from these studies was extracted into a customized Microsoft Excel sheet. Qualitative assessment of the included studies was performed using the QGenie tool, which is specific to assess genetic association to the phenotype of interest. Quantitative synthesis was performed at the SNP level if the genotype frequencies of the SNP were reported in various studies. To better understand the effect of polymorphisms across the gene, a gene-based analysis was performed, and to comprehend the effect of polymorphisms on the biological processes, a gene cluster analysis was conducted. Two bioinformatic tools, that is string and gene mania, were used to understand the effect of the polymorphisms on the susceptibility to ECC. This slide depicts the Prisma flow diagram, and it can be seen that 15 studies were included into the systematic review, and 2 studies which reported the genotype frequencies of a single SNP were included into meta-analysis. The qualitative assessment was conducted using the QGenie tool. Totally 15 studies were included into the systematic review, of which 14 studies had low risk of bias or were of high quality, and a single study had moderate risk of bias or was of moderate quality. The quantitative synthesis was conducted if the genotype frequencies of the SNP were reported. The forest plots generated revealed that the wild homozygous genotype and the mutant homozygous genotype of a SNP in the beta-differentiation gene had an odds ratio greater than 1, but with insignificant p-values. To overcome the shortcomings of including only 2 studies into the meta-analysis, a gene-based analysis was performed. This gives a clearer idea on the effect of the polymorphisms across the gene on the phenotype of interest, and the results revealed that 2 genes, that is T receptor alpha variable 4 and tumor necrosis factor alpha, were significantly associated with ECC. Certain statistical analysis revealed that certain polymorphisms in lipoxygenase 15 gene were also associated with ECC. In contrast to the findings of the gene-based analysis, gene cluster analysis did not reveal any statistically significant association between the polymorphisms of these genes and ECC. To better understand the effect of the polymorphisms on the biological processes, a string plot was constructed using the string database. String stands for an acronym, meaning search tools for retrieval of integrating genes. The string plot constructed reveals 9 nodes representing the genes and 8 edges representing the interaction between them. These genes were actually clustered into 3 groups. Group 1 consisting of 2 genes, that is mannose binding lectin 2 and mannose binding lectin associated serine protease 2. Group 2 consisting again of 2 genes, LPO and LTF, that is lactoperoxidase and lactotransferrin, and Group 3 consisting of 5 genes, lipoxygenase 15, human necrosis factor alpha, beta-defensin 1, and human leukocyte associated allele DRB1 and DQB1. Further analysis of the string plot revealed that 7 genes were associated with humoral immune response, and 4 genes within those were associated with humoral immune response mediated by circulating immunoglobulin. 6 genes were associated with innate immune response and 2 genes among them with innate immune response of the mucosa, and 3 genes with regulation of phagocytosis. The gene mania plot constructed shows the genes arranged in 2 concentric circles. The genes in the inner circle represent the genes of interest, and the genes in the outer circle represent the interaction between these genes. And it is very clearly stated that the physical interaction between these genes is to an extent of about 39%, and co-expression of these genes is pretty common to an extent of about 34%. Coming to the discussion, interaction between the genes regulating the immune response of the host and bacterial genome paves the way for susceptibility to any disease. Polymorphisms of immune response genes alter the defense response of the host and can either facilitate or inhibit the colonization by karyogenic bacteria. Polymorphisms of the genes can occur either in the exonic region or the intronic region. Those which occur in the exonic region alter the protein, and those in the intronic region affect the transcription. However, wherever the polymorphism is located, it can have a minor effect. So to better understand the effect of these polymorphisms across the gene, a gene-based analysis was conducted. And to better understand the effect of these polymorphisms on the biological processes and on the functional grouping of genes, a gene cluster analysis was performed. The results revealed that the levels of beta defensin 1 and human necrosis factor alpha were higher in salivary samples of children with ECC, and genotype TT of a SNP in the lipoxygenase 15 gene was reported to be a risk factor for ECC, whereas the heterozygous genotype of a SNP in lactotransferrin gene was reported to be protective against ECC in the Turkish population. Gene-based analysis revealed that the polymorphisms across the T receptor alpha 4 variable and tumor necrosis factor alpha were significantly associated with ECC. The enrichment analysis was performed using the string tool and the gene mania tool. The string tool, which is search tool for retrieval of integrating genes, revealed that the biological process of lectin pathway of complement activation and the humoral immune response and the innate immune response of the mucosa were statistically associated with early childhood caries. Gene function prediction using multiple associated network integrated algorithm revealed that these genes were physically interacting with each other, and the way that co-expression among these genes was pretty common. So to conclude, polymorphisms of tumor necrosis factor alpha, T receptor alpha variable 4 and lipoxygenase 15 and human leukocyte antigen LDRP1 can modify the susceptibility to ECC. Also, the genetic variants of genes regulating the lectin pathway of complement activation can modify the susceptibility to ECC. Interaction between these immune response genes and the bacterial genome is dynamic, and one really needs to understand the antics of these genes to better understand the genetic underpinnings of early childhood caries. These are my references. I would like to extend my heartfelt gratitude to the organizers of CECCRe 3.0, to my guide, Dr. M.S. Muthu, my co-guide, Dr. Vetriselvi, and to my RAC member, Dr. N. Sivakumar, and to my family for their constant support and encouragement. Thank you.